Well, hello. My name is Dr. Ken Peters, and I am a petroleum geochemist with interests in basin and petroleum system modeling. And I'm currently a co-principal in the Stanford University Basin and Petroleum System Modeling, or BPSM, Industrial Affiliates Program. We've decided to prepare a short lecture series, condensed lecture series for our students, our PhD students in BPSM, that reviews some petroleum geochemistry for basin modelers. This is also going to be made available to our co-principals, but we also have decided to make this available to our industrial affiliates in the program. We certainly value their contributions to the program. And start. This is a figure that I prepared in a paper from 2008, and it's kind of a thumbnail sketch, at least of my view, the way a basin and petroleum system modeling actually maps out. The importance here of this slide is that it demonstrates that geochemistry is really absolutely critical for a good basin model. Geochemistry provides both the input and the calibration for these models. Here, for example, we have geochemical input, organic richness, that's total organic carbon, things like the hydrogen index, the oil or gas-prone character of the organic matter, kinetics of that organic matter, that is how fast it reacts during burial maturation. And also, there are other geochemical inputs related to some of these other things like the boundary conditions and so forth. Now, basin and petroleum system modeling really consists of two sort of parallel reacting simulators. One simulates the geohistory of the rock. The other simulates the geohistory of the fluid, and a long way of describing that is basin and petroleum system modeling. A lot of people use the short term, either petroleum system modeling or basin modeling, and I may do that myself. But we basically are calculating two different things here. And of course, we generate then a model. However, that model is uncalibrated. It's uncalibrated. Now, there is a very famous statement out there in the literature from a statistician. His name was George Box. And he made the statement, all models are wrong, but some are useful. Of course, every basin model is ultimately wrong because the amount of detail in the geohistory of a basin is almost impossible to replicate, but we do want to generate a model that is useful. It's at least a good approximation of what has occurred through geohistory. And the best way to do that is to calibrate this model. And that's done, again, with a lot of input, a lot of calibrants. So for example, here's a geochemical calibrant, vitrinite reflectance. We can also calibrate against corrected bottom hole temperatures, for example, or pore pressure, or many other parameters that we can measure in the field and compare to what the model predicts. So we can iterate multiple times if necessary. As we get more data, we can iterate again and improve the fit of the model to the actual measured data at various locations within the model. So this is a very powerful technique, and it generates models that are useful. They're useful in terms of prediction, and they give competitive advantage to those who use them properly. Now, I want to start off the lecture here with a short definition. The term petroleum system is used by a lot of people in our industry. Unfortunately, it's probably one of the most misused terms that we deal with. It's actually quite rigorously defined. Les Magoon and Wally Dow define it quite nicely in their 1994 book. There are 4 essential elements and 2 processes in a petroleum system, and that consists of all the related petroleum that originated from one pod of active source rock, that is thermally mature source rock. So here are the essential elements: source rock, reservoir rock, seal rock. Of course, all of these have geochemical characteristics that we can measure. And the underburden is also an essential, the overburden, sorry, is also a very important element. And that's because this is what buries the source rock to great enough depth to generate the petroleum. Now, there's also processes that are considered in the petroleum system concept, trap formation and generation, migration, accumulation. And of course, timing here becomes an issue. If the trap forms after the generation and migration, then the generated hydrocarbons may not be trapped. They may migrate and become dispersed or escape to the surface. So timing is an important issue here, and that's a geochemical issue as well. That's involved with the kinetics of the organic matter in the source rock and how it responds to the geothermal gradient and the burial history that you have built into your model. Okay, well, here are the 6 chapters that we want to describe to you in the next lectures. And we're going to break these into separate lectures so that each of you can go and see a lecture and isolate one lecture and see that lecture and think about it. And then when it's convenient, come back and look at another lecture. So this is sort of the sequential order of those lectures. We're going to start off with a discussion of the origin and preservation of organic matter. So there's some basic concepts here that many modelers may be aware of, many others may not, but I think we need to cover those to build a foundation for what we go on to later. Vitrinite reflectance and rocky valve pyrolysis TOC. These are workhorse fundamental tools that are used as input and calibrants in basin and petroleum system modeling. Many geologists and even geochemists think of these as simple and easily interpreted. Not so. These are very powerful tools, but the interpreter needs to be aware of the pitfalls to interpretation. And there have been some very new developments, recent developments in vitrinite reflectance, the kinetics of vitrinite reflectance, for example, and in pyrolysis that people need to know about. We'll talk then about kinetics. That's also a very important topic. Just as an example, I've had students who have prepared wonderful 3D models with all the input and put in all the time to build the input. And then they push the button on the software and the software comes back and says, no, we can't run this. You haven't given us kinetics for the source rock. So then the student goes and in a rush selects some default kinetics from the software, which may or may not have the same kinetic parameters as the kerogen in their particular source rock in their study area. So kinetics is a very important topic. There have been some new developments there that we want to talk about too. These are very recent developments. We want to talk also about geochemical correlation. This is something that many basin modelers simply don't think about too much, but I can tell you, I've seen publications in the literature of full 3D models where the authors got the source rock wrong, or they missed a source rock. And this, of course, means that that model that they've prepared, all the work that they put in, is maybe not that useful. So every geologist who does basin modeling needs to know about geochemical correlation. They may not actually do it, but they need to be able to evaluate the quality of the geochemical correlations and the established petroleum systems that are established using those correlations in terms of what they actually put into their model. And finally, we're going to finish with a little lecture on gas geochemistry. A lot of developments there recently, especially along the lines of biogenic gas generation and gas hydrates. So we will talk about that. There's some really new stuff there that needs to be discussed and made available to our students and to our affiliates. So I'm going to read this little section here down at the bottom because I think it's important. I want to emphasize this. To be useful, predictive basin modeling must include supporting input and calibration based on petroleum geochemistry. This short refresher course focuses on fast geochemical screening techniques and some more detailed geochemical methods that contribute to useful basin models. Basin modeling without sound geochemistry is a recipe for disaster. So that's I think, very, very true. So let's start off with a discussion of the origin and preservation of organic matter. To do that, we have to talk about the domains of life. Up until just a few years ago, really, people were not aware of this entire domain here, the archaea. We call them archaebacteria or the archaea. These are an unusual group of organisms. They include extreme halophiles. They live in very saline environments. Methanogens that can generate methane from organic matter. Extreme thermophiles that can live at very high temperatures. Some of these organisms are reproduce best at autoclave temperatures of over 100 degrees centigrade. So here's some methanogens over here on the right, photograph of them. The cluster distance you see here in this dichotomous phylogenetic tree is based on similarity in the 16S rRNA, okay, the ribosomal RNA. And this that RNA is found in virtually all organisms. So this can be sequenced. We have now wonderful instruments that can sequence the genetic code in the RNA, and then we can make comparisons, statistical comparisons, that actually reflect in the cluster distance between these different classes of organisms. And you can see the archaea are distinct from the bacteria that we're more familiar with. Here, for example, green bacteria. Here's Spirogyra, which is a green bacterium. Now the eukaryotes, of course, these are what some people call higher organisms. Well, I don't know, these are all basically higher organisms when you think about their environments that they live in. Plants, fungi, animals, people too, as well as cellular slime molds. So, these are organisms that have a distinct nucleus, that's a nuclear bound membrane around that nucleus, and they have the DNA in the nucleus. Here's an example of a eukaryote. This is actually an organism I worked on when I was a very young student. This is Volvox aureus, which is a multicellular alga. They're these little spherical organisms that consist of multiple cells, each of which has flagella. And this organism is phototactic. And you can see it's green, it's got chlorophyll in there. It's phototactic, which means these little sort of basketballs are very small, but they move toward the light. They're phototactic because they want to use that light to fix carbon. And we'll talk about photosynthesis and fixing carbon in a minute. Well, this is a dichotomous phylogenetic tree. You can also depict this in another way, which is with a fan-shaped phylogenetic tree. Again, the cluster distance, or the distance here between types of organisms, is indicated in terms of the difference between the 16S rRNA sequences. On this slide, though, I've added some extra items here. These different types of organisms, these different domains have many, many different types of species within each of them, and they all generate different types of what we call biomarker precursors. A biomarker is a compound you find in crude oils and in source rock extracts, and biomarkers can be traced back. They're biological markers. They can be traced back to a component in a once living organism. So let's look at the bacteria, the eubacteria here. And I just included a few examples of some classic biomarkers. And the idea here is to introduce some later discussion, a little bit later discussion we'll have on biomarker analysis. This, for example, and this compound, these are pentacyclic triterpenes. They're very complex structures. They come from basically cellular membrane components in the bacteria. The hopanoids. This is a 2-methyl hopane. That's a 3-methyl hopane. Whoops, 2-methyl. Well, I've got a 2-methyl and 3-methyl hopanes here, and these are right away, you see these in a crude oil, you know that there were bacteria in the source rock. Here's a compound that is found in eubacteria. It's, look at this here, it's isorenurotane, okay? And here's another one with a similar structure. It's called chlorobactene, sorry. It's an oral isoprenoid. These sorts of compounds are manufactured, or their precursors are manufactured in the kinds of organisms that we're talking about here. Now we can go over to the eukaryotes, and of course they actually have a different composition in their cellular membranes. They use sterols, and you see a lot of sterols here. These are products of sterols. This is the C29 stearine, yeah, and it's also called stigmastane. The popular name is stigmastane, and it's common in sort of higher plants, but it's also found from other organisms as well. Here are two C30 stearines. You see they differ a little bit in the structure that's attached here, the carbon number 24. This one right here is called 24N-propyl. It's got 3 carbons attached to the 24 position. 24N-propyl cholestane. And that is a marker of chrysophyte algae. The precursor is found in chrysophyte algae, which are marine organisms. So if you find this compound in your crude oil, you can infer that the source rock was deposited under marine conditions. Okay? Here's another one. This is a very interesting stearine. It's a C30 stearine also. It's a C30 isopropyl cholestane. So there's an isopropyl group attached here. This is a rather unusual structure, and the precursors for this are found in sponges, porifera. Okay, and if you look at the ratio of this compound to say that compound through geologic time, there's a spike, a very high abundance of these compounds back in the early Cambrian and the late Vendian, the lattermost portion of the Precambrian Proterozoic. So this is actually not only a source biomarker, but it's also a biomarker that can be used to say something about age. If you have a lot of this in your oil, it's likely that it's from the sort of the Infracambrian time period. Okay, here's another biomarker that's common. There are many of these. These are diterpenes, and there are many of these in higher plants, for example. Now down here for the archaea, these are, again, there's lots of very interesting organisms here. Methanobacterium, Methanopyrus, Methanococcus. A lot of these make methane. Here's Halobacterium, which is a very, very tolerant of high salinity. Here's Pyrodictium. This is a very interesting organism. It actually reproduces best at about 110 degrees centigrade. And of course, that's under pressure, but so the water is liquid. But these organisms have unusual cell membranes, and those cell membranes, of course, can sometimes be preserved in the sediments, and the precursors then are converted into products like this compound right here, which is a cell membrane component, at least its precursor is. This one is called biphytane. Some people still call it bisphytane. Biphytane. It's a C40 head-to-head isoprenoid, and you can see it was constructed by a living organism because it has these repeating subunits in it. They're called isoprene subunits. And in fact, all of these compounds are built from isoprene biosynthetically. Here we have another compound, and this would be, if I'm correct, this would be crochetane. And this is a very unusual compound too. It colutes with phytane, which is a big peak on most gas chromatograms. We'll talk about gas chromatograms in a little bit in one of the next lectures. But phytane comes from chlorophyll, This thing, this crochetane, is a marker that sometimes interferes with the phytane peak or can contribute to it because they coelute. They come out at the same time from the gas chromatogram. And this is a characteristic of anaerobic methane oxidation. So the organisms that make this are taking methane and oxidizing it to CO2. So you can see just from this little taste of what I've given you here of these different types of organisms and the different kinds of input that they have, we can tell a lot about the depositional environment of the source rock for an oil, even if all we have is a crude oil sample. So that's very important. We want to establish that link between the crude oil or the fluid and the source rock. And that's what establishes the petroleum system. We'll come back to this a little bit later in later lectures. I want to come back now and look at sort of biopolymers. These are the major components that are deposited in sediments by living organisms. And I've got just a few examples here from John Hunt's book from 1980. And here we have some higher plants: spruce, oak, Scots pine. You can see they contribute all of these biopolymers. Protein, for example, your hair is protein. It's peptide linkage of amino acids. Yeah. And carbohydrates. Carbohydrates, these are basically sugars with glycosidic linkage, sugars like glucose and fructose and galactose. They're bound together to make carbohydrates. And these, of course, are easily broken down. These glycosidic bonds and the peptide linkages are easily broken down by microorganisms in the water column and in the sediments. So these types of biopolymers really don't fare very well during deposition, during diagenesis. We'll talk about diagenesis a little bit more, but basically I'll define it here. It's everything that occurs to the organic matter during deposition and immediately after deposition before significant thermal maturation, before, say, 50 degrees centigrade. Okay, so degradation of these biopolymers is extensive. Also, lignin is sort of bound the same way as carbohydrates, and it's also susceptible to early degradation during diagenesis. Lignin you find in higher plants. It's the component that allows them that ability to stand up, basically. A tree requires lignin as a structural component to help make it a rigid structure. Lipids are a little bit different. Lipids are, of course, biopolymers. Most of them are produced, well, virtually all of them are produced by polymerization of isoprene in various ways. And lipids consist of things like fatty acids and waxes and pigments, sterols, hopanoids. The interesting thing about these compounds is they're really the backbone or the skeleton of these compounds is carbon to carbon covalent bonds. So those are very difficult to break compared to peptide linkages or, glycosidic linkages. Carbon-carbon bonds are more difficult to break. So lipids, of course, have peripheral groups when they're in an organism, like a fatty acid has an acid group, carboxyl group basically attached to the end of the chain, but that's readily broken off during diagenesis and you end up with a straight chain, maybe a straight chain alkane. Okay. So lipids can undergo preferential preservation because of their structure, covalent carbon-carbon bonds. You see here in the phytoplankton, the diatoms, the zooplankton, lots of carbohydrate, lots of protein, no lignin, of course, but they also make a lot of lipids. So those lipids can be preserved, and we can see markers of diatoms and markers of different types of phytoplankton and markers of various higher plants, biomarkers, in sediments and in crude oils that are derived from those sediments. Now, I do want to spend a little time on this figure. This is something that people want to keep in mind when you think about what rocks you're going to include as source rocks in your study. This is this is the redox cycle for carbon on Earth, and this is a cycle that's been established for billions of years. A very important part of this was the evolution of chlorophylls and other pigments that allow plants like algae and cyanobacteria and phototrophic bacteria to do photosynthesis. Photosynthesis, we take carbon dioxide and water, and we use light energy to convert those then to biomass with oxygen as a sort of a toxic byproduct, right? Most people don't think of oxygen as toxic, but it was very toxic to the early organisms on this planet because our atmosphere contained no oxygen originally. It was probably rich in carbon dioxide, but virtually no oxygen. In fact, we know that because iron, prior to a couple billion years ago, was dissolved in the oceans. It was soluble. It was the ferrous form of iron rather than the ferric form of iron. Same with uranium. We have this photosynthesis going on, aerobic photosynthesis and anaerobic. The first photosynthesizers were anaerobes, of course, and that means they don't use oxygen down here. Okay, so an anaerobe can take the organic matter, the organic matter that's been produced by these photosynthesizers here, and break that organic matter down by either respiration using something other than oxygen as an electron acceptor like sulfate, or they can ferment that organic matter and break it down, get energy for metabolic processes and generate CO2, okay? So this is a very efficient cycle, yeah? And so is this one, the aerobic cycle, which came around later. We have organisms now that use oxygen instead of sulfate or other things for respiration, right? You and I are on this arm of the aerobic cycle here, where we use oxygen to push this reaction to the left. We take oxygen and organic matter, and we exhale carbon dioxide and water. So this is another remarkably efficient cycle, very, very efficient. Almost all the organic matter that is synthesized by primary productivity, fixing that carbon into that organic matter, almost all of it is recycled, okay? There's a book by Bernard Tissot and Dietrich Welty that many of you are familiar with. And in that book, they state that about less than 1/10 of 1% of the primary productivity is preserved in sediments. So there's a tiny leak here and over here where that organic matter escapes the cycle and is buried in the sediments. Tiny leak. And it's occurred for billions of years. And in fact, that leak, if you think about it, is pulling this reaction to the right. We're burying organic matter and we're generating more and more oxygen. Eventually, the oxygen content rose to the point where we were oxidizing the ferrous iron in the water column in the oceans, precipitating, the banded iron formations, precipitating, detrital uraninite. So the present day atmosphere is 21% oxygen, okay? And that's because this reaction has been pulled to the right due to burial of organic matter by these tiny leaks that have occurred. This accounts for all the coal and all the kerogen. There's another term, kerogen. Most people know what that means. It's the particulate, insoluble organic matter that you find in sediments. And that's by far, the dominant form of carbon organic carbon on the planet. So that's the redox cycle, and it plays into, of course, source rocks. Here we have land plant organic matter. This, of course, is since the Silurian, and aquatic organic matter being deposited in usually fine-grained sediments here. Okay? And the deposition can occur under oxic conditions, where the water column contains, say, 8 milligrams per liter of oxygen, or it can be under anoxic conditions where you're less than 0.5 milligrams per liter of oxygen, or zero, near zero. That's anoxic. Different types of organisms live in these environments. These would be aerobes, and these are anaerobes, as I just showed you in that previous slide. And if you have enough organic matter that is preserved under these conditions, you have a potential source rock with good total organic carbon and the potential to generate oil and or gas. Now, then this is buried and it can become an effective source rock if the temperature and the geothermal gradient is adequate to do this. And we can generate and expel hydrocarbons that are trapped under seals, or you know, the unconventional targets that we have today. There's a lot of that organic matter that stays in the source rock and is still there and available. So what we're trying to do in basin modeling is to simulate this, yeah. And this other part of the figure from the Biomarker Guide, it's one of my books, is kind of a description of these terms: Diagenesis, Catagenesis, Metagenesis. If anybody uses the term immature oil, they really shouldn't do that. Oil by definition has to be mature. So immature is by definition diagenesis, you know, less than say 50 degrees centigrade. We do get some petroleum. We get biogenic gas at low temperatures, methanogens, and make methane, basically pretty pure methane at very low temperatures. The difficulty here, of course, is trapping that methane because seals and competent seal rock is not formed yet. But there are mechanisms for trapping this, like salt deposition or permafrost or gas hydrate type clathrates. Of course, the biomarkers are, you know, are useful. That's what this book is at least partly about. They're useful because they're quite stable thermally, and they're stable, quite stable, resistant at least to biodegradation. And of course, we get to high enough temperatures, we generate oil, the oil window. Eventually, the kerogen becomes depleted in hydrogen, and we start to run out of the oil generative capacity, and we generate just lighter and lighter product, wet gas, and then dry gas or gas condensate. Thermogenic gas, we'll talk about that in the last chapter that we discuss, the lecture 6. You can tell the difference between biogenic and thermogenic gas with composition and isotope data. So that's kind of a background slide. This is a thumbnail of what we're going to be talking about later. I want to talk now about some, I think, a fundamental paper that was written by Jack Middelburg back in 1989. And Middelburg was concerned with looking at the relative rate of the degradation of organic matter with time in sediments. So he actually plotted up a log of the rate, yeah, the log of the rate, right, as a function of the log of the time, and he got a straight line, which to me, I still find this an amazing correlation. This is amazing. It doesn't matter what type of organic matter it is. There's a pretty linear function here that says, as you get deeper and deeper, there's less and less organic matter. And it's being basically destroyed as this rate constant changes. It's not really a constant, it's a rate, right? And so he came up with this equation to fit that line. And he used two terms here. This is the time term, that's the age of the sediment. But he found it wasn't just simply the age of the sediment that determined this reactivity of the total organic carbon. It was also this factor right here, and some people have overlooked this. This approach can be modeled, can be used in models to predict, for example, methane generation from methanogens in shallow sediments. But some of the papers that have come out have actually ignored this term right here, which is a very critical factor. He calls this the apparent initial age, okay? Petromod version 2018.1 has a special option where you can include that value of A. So it's very important that that be included. The first 3 columns here on the left of this table are from Middelburg's paper. You can see he's listed some depositional environments here, present day depositional environments, and some corresponding papers. Now, I know these papers, and I looked at these and I said, well, gee, you know, Saanich Inlet and Cape Lookout Bight, these are anoxic. They're notorious for being anoxic depositional settings. No oxygen in the sediments, okay? On the other end, we've got the Central Pacific, or the Hatteras Abyssal Plain. These are extremely oxic sediments, and the water column above them is oxic. Very little organic matter ever gets to the bottom in the middle of the Central Pacific, or the Hatteras Abyssal Plain. It's all oxidized before it gets down there. Here you get pretty good preservation of organic matter in those sediments. And then intermediate is here. So I've added these redox indicators, approximate redox indicators here, from anoxic to oxic. And look at the difference between the apparent initial age for an oxic sediment. That's a big number to substitute into this equation. So that has a major effect on your reactivity of the total organic carbon in your model for, say, methane generation. Okay, so let's talk a little bit more about this redox situation here. This is the typical situation that you see present day around the oceans and the lakes around the earth. There are exceptions. We'll talk about those in a minute. But most are oxic settings. And this is from a paper by Gerard de Maison and Moore from 1980. And so in an oxic setting, here's the water column. And say you're a diatom, and that diatom dies and starts to settle down through the water column. You know, it's going to take days or months for that diatom to reach the bottom. In the meantime, it's going to be fed on by a whole variety of different organisms. So it's going to go through the guts of different organisms on the way down for months. And then once it reaches the sediments, it's still going to be in the presence of oxygen. The interstices between the silt grains or the mud, the pore spaces between the muds are going to contain oxygen. Why? Because this sediment is being bioturbated by metazoa, multicellular organisms like worms and crabs. And so this is bad news for that organic matter. It's largely going to be destroyed. And this could go on for hundreds of years, okay? Hundreds of years down here. So these sediments in the future, if they're preserved, these sediments will be massive. They will show basically completely bioturbated sediment. Nearly all of the organic matter will be decomposed. And the typical values are around 0.2. In the middle of the Pacific, they're less than that, 0.1 or less weight percent TOC. You do get some values up in the 3, 4, and even 5 weight percent TOC range. You can imagine if you buried this very, very quickly, say at in a delta, like the Niger Delta or the Mississippi Delta, that organic matter could be removed from this oxic zone a little more quickly with very rapid subsidence and deposition. Once you get out of that aerobic zone, there's another consortium of organisms. There are different organisms at all these levels. Another consortium of organisms, these guys do sulfate reduction, for example. And this is a much less efficient, sulfate is a much less efficient electron acceptor than oxygen. These guys are very efficient at destroying the organic matter. Sulfate reduction is less efficient than that. And in destroying the organic matter and making it into CO2, you make hydrogen sulfide. That's that rotten egg smell that you sometimes will experience in lagoons and so forth. So that's the sulfate reduction zone. And then with further burial, you run out of sulfate and these methanogens start to take over. Now, methanogens can't compete with the sulfate reducers and certainly can't compete with the aerobes. The methanogens are, of course, poisoned by H2S, as are the aerobes, and they make methane. And they make this in 2 different ways that we'll talk about when we talk about methane or biogenic gas generation. CO2 reduction, we'll take the CO2 and reduce it with, say, hydrogen to make methane, or acetate fermentation, where they'll take a substrate like acetate, and they'll basically ferment it, break it apart to make methane. Okay, so that's oxic settings, low TOC, not very good preservation, and in general, pretty low quality of organic material. Now, in the geologic past, and even some places today in the ocean, and certainly in some lakes, we have anoxic settings. Again, anoxic settings, the the oxygen content is near zero, and we can get very good preservation. In this case, the example I'm showing you here we have is, it's oxic in part of the water column, but then it goes anoxic in the water column. Typically, this dashed line is represented by a thermocline or a halocline. This might be more saline than the above water. And so these two water bodies have become isolated. And as this organic matter drops through the water column, there is biological oxygen demand. The oxygen is being used up to oxidize that organic matter, and eventually you run out of oxygen, then you're into the anaerobic zone, and you get things like bacterial sulfate reduction here. Even in the water column, that can occur. Once you get to the sediment, that organic matter is again subject to degradation, but now you have no aerobic metazoa, no burrowing worms or clams, or there are no metazoa that are churning the sediment up. So you end up with laminated sediments. If you look closely enough in virtually every source rock, you will find evidence of lamination. Usually this is a very fine lamination on an annual or even sometimes semi-annual basis with varves that record an algal bloom and then more siliciclastic, say, deposition, and then another algal bloom in very sequential pattern here. And so this is in the future going to be a laminated, organic rich shale or carbonate that preserves high total organic carbon and also probably better quality organic matter in terms of making oil versus gas. 25% or even more organic carbon. And you can continue burial and get back down into the methanogenic zone. This may take hundreds and hundreds of years, and of course, this degradation is much less efficient. Now, you see this basically in a sewage treatment plant. A sewage treatment plant, the organic matter is so abundant that there's heavy biological oxygen demand. It's anoxic. So in sewage treatment plants, the technology is basically to pump in air, pump in oxygen, and that pushes the organic matter to be degraded then to CO2 and water and mineral remains. Okay, so we can look at this in a little more detail. This is now, we can see some straight lines that are defining these zones. Well, the zones are actually a little fuzzy. They're actually a little fuzzy. Remember, there are different consortia of organisms, multiple populations of different types of organisms in each of these zones that we've defined here. And these zones are defined basically by the electron acceptor. Here, oxygen, very efficient at taking that organic matter and breaking it down to CO2 and water using oxygen, right? In the suboxic zones, and the sulfate reducing zones, we're using another type of electron acceptor. Sulfate reducing zone is very common because there's a lot of sulfate in the ocean. And so you take the organic matter, instead of using oxygen, you use sulfate to make basically CO2. And H2S, which is that toxic gas. We have the methanogens down here, and they're deeper than the sulfate reducers. They can't compete with those. H2S is toxic to them. So, this is a boundary that's defined, at least in part, by that. These guys are making methane by acetate fermentation or CO2 reduction, but that methane, if it moves up, is going to be maybe oxidized. This is basically reverse methanogenesis. This is methane, sulfate reduction of methane. Okay, so we're destroying that. So that kind of defines this boundary more or less. And then there's this boundary down here. Where do the methanogens cut out? And how can we predict where the methanogens will be most accurate? Well, most basin modeling programs try to do that just with temperature because these guys are temperature sensitive. Yes, they don't like temperatures over about 80 degrees C, for example. But, there are other things involved here in making methanogens happy. For example, they need a substrate. They need acetate, or they need CO2 and hydrogen. to work. If the sulfate reducers and the other organisms above them use up all of the acetate, they can't do fermentation. So there are other limitations besides. It's not as simple as just temperature dependence here. So this boundary down here is a little fuzzy because of that. It's not just the temperature that you basically make them go dormant above a certain temperature or you kill them. There's also substrate availability and pore space and other things, presence of H2S, other things to consider in terms of what limits the methanogens. We'll talk about that in the gas lecture, lecture number 6. Okay, I want to show you some photographs. This shows a typical Oxic setting. This happens to be 607 meters in the Gulf of California. And you can see this sort of smooth, hummocky appearance of the sediments with all these filter feeders and worms extending the little filter feeders. They're basically trying to capture organic matter that's floating with currents along the bottom here. And there are a lot of bioturbation. There's a lot of bioturbation going on here in the subsurface. That's because these are aerobes. They're very efficient at destroying the organic matter. Millions of years from now, this sediment will be a massive sediment. Now, I've had students ask me, well, can a siltstone or a sandstone be a source rock? Well, can a mudstone be a source rock? Yeah, sure, a mudstone can be a source rock, but it gets more tenuous when you deal with siltstones or with sandstones, because those are deposited generally in oxic settings, and the bioturbation and the oxygen content during deposition is going to destroy the organic matter. So this is a setting that is probably not going to be a source rock in the future. Here's sort of an intermediate setting. It happens to be deeper, but you've got to be real careful about just applying depth in the water column as a criterion for selecting the oxicity. You can have oxic at the surface, anoxic at some depth range, and then back to oxic again. So this is dependent on a lot of parameters besides just water depth, like water circulation and biological oxygen demand and so forth. But this is kind of an intermediate situation. We have some oxygen present, but it's very low. It's approaching 0.5 milligrams per liter of oxygen. And you see the organisms are stressed here. There are really no surface organisms. We've got some burrowing organisms. So in the future, millions of years from now, you might see this sediment. You may see some laminations in the sediment and then a burrow, okay? And then some more laminations and then another burrow through those sediments, okay? Now, this is another location. This is the Black Sea from the Atlantis II cruise. And you can see here, it's about 1 meter across. There's virtually no character to the bottom sediments. There are no surface mounds or burrows. These sediments are basically intact. If you dropped a coring device into this, you would find that the sediments are finely laminated, recording annual varves. Yeah, there are no aerobic organisms here in this bottom sediment. In fact, in the overlying water column, it's sulfate reducing. It's anoxic or suboxic at least. And this means good preservation of organic matter. You've eliminated the bioturbation. And this has some interesting side effects. This is actually a Byzantine or an Ottoman ship in the Bulgarian portion of the Black Sea. And that's about 1300 AD. And you look at that ship, the wood is still pretty much intact. This sank in a storm, and at the bottom there are no aerobes to eat the cellulose in the wood or the lignin. Okay. Now, in fact, Robert Ballard of Woods Hole Oceanographic has done a lot of exploration with submersibles in this part of the world because of this depositional environment, and they found triremes from the 5th century BC. The ancient Greeks had these triremes, or battle ships, basically, with lots of oars, with oarsmen, and then a big metal prow to ram into other ships. 5th century BC trireme, you can still see the wood. You can still see the structure, the way the trireme was built. So that's an anoxic depositional setting with very good preservation of the organic matter. Now we can also consider one of the other variables that I talked about just a minute ago, and that is sedimentation rate. In an oxic setting, if you look at sedimentation rate, yes, it has a major effect on preservation of organic carbon here. It's a log scale here, and you can see here these dots are normal sort of terrigenous dominated organic matter being deposited in deltaic settings, sort of deltas. And you can see that if the sedimentation rate is high, you can get better preservation, 3, 4, 5% TOC. This is from Régis Pellet, very nice data set. So the level of preservation increases, in some of these deltas, like the Akbara formation and then the Niger Delta. TOCs get up 3, 4, even 5%. Some of these the stars here are areas of high productivity. For example, off of Namibia in West Africa, there's some high productivity. You may have deltas nearby, but you're also getting organic matter deposited from these highly productive waters. Now, there's also some evidence that this trend will turn over at very high sedimentation rates. Most sedimentation rates are sort of in the range of 1 to 10 centimeters per 1,000 years. You get up here to very high sedimentation rates, you're going to dilute that organic matter with minerals. Now, the interesting thing is that under anoxic conditions, it doesn't really matter. It doesn't really matter what the sedimentation rate is. Most anoxic settings, of course, are quiet water, isolated water body, so the sedimentation rates are lower. But because the water is anoxic, or the sediments are anoxic, the preservation is about the same. And it can be quite good. It doesn't have to be 10%. It can be more than this. Okay, let's look at an ancient example. These are the Liassic shales from Yorkshire. And in the same shale, if you move laterally through that shale, you can find the different shale facies that have different kerogens in them. So here is the normal shale facies. It's dominated by, basically type 3. We'll talk about kerogen type in a bit, but it's gas-prone kerogen. The hydrogen index is fairly low from Rocky Val. Okay, it's a low hydrogen index, so it's going to be a gas-prone kerogen. And TOCs are not particularly high, okay, 0.6 to 3.4%. And if you look at the trace fossils, the bivalve groups, you've got lots of suspension feeders, infaunal and epifaunal species, lots of chondroides trails and horizontal burrows. And this is sort of the environmental interpretation. Oxic water bottom, oxic in the sediments down to some depth where then we get into the sulfate reducing zone. But that's going to really bioturbate those sediments. The TOC is going to be low because of that. The quality of the organic matter will be low because a lot of it's being oxidized. At the other end, other extreme here is the bituminous shale, still within the Liassic shales here. And here we have type 2 or oil-prone kerogen. See the hydrogen indices. Type 2 kerogen is more or less defined as hydrogen index between 300 and 600 milligrams of hydrocarbon per gram TOC. These are basically type 2 oil-prone. And look at the TOCs, 5 to almost 12% total organic carbon. And we don't see, a lot of burrows, and no burrows. There's maybe some epifauna or suspension feeders, but very, very limited. So we probably had a situation very similar to this. Anoxic bottom water, and then the sediments, of course, below that even are anoxic as well. Okay. Now these are some spectroscopic observations from the NASA's mission to planet Earth from 1997. And they're focusing in here the sensitivity with a spectroscopic observation on chlorophyll. Basically, where's the chlorophyll? Where's the most phytoplankton? Where's the most active populations of phytoplankton? And it's very interesting. You can see tremendous blooms of algae along the west coast of continents. Yeah, down here in Peru, Some of these sort of extend way out into the ocean. Yeah, these anoxic zones that correspond to that. And here offshore Namibia, the western side of the continents, well, you're getting Coriolis force here in the northern hemisphere. You're getting Coriolis force. It's a shallow water phenomenon, but it brings up nutrients from the sediments from about 200 meters or so depth. And those nutrients are washing off the continents. And you get these tremendous algal blooms. For example, I'm from California, and we get every few years we'll have a big red tide. They call it red tide. And it's actually pretty much a monoculture of an organism called Gonulax polyhedra. And it makes the tide, the water look red, and also the filter feeders, the clams, the filter feeders pick this up, and it doesn't seem to bother them, but Gonulax makes a very potent toxin. And so you can't eat the shellfish certain times of the year along the west coast of these continents because of these blooms, and they contaminate the shellfish, and you can die from eating that. The other thing to note in this figure is the tremendous bioproductivity up here in the Arctic and down in the Antarctic. You see places like the center of the Pacific Ocean here are really kind of deserts. Here's Hawaii. Yeah, there's some good productivity right around the Hawaiian Islands, but if you get away from those islands, there's very poor productivity in the water column. Okay, so there's a lack of nutrients here, and the organisms, the phytoplankton, simply can't do their thing. So here's where all the activity is, right? And now what I'm going to do, well, you can see it's on land too, here the Amazon forest, but in the ocean is what we're mainly concerned with in terms of source rocks. What I'm going to do now is go all the way back to 1980, and this is this Maison paper I showed you, a remarkable paper from 1980. You see it looks very similar to what I just showed you. This is milligrams of carbon fixed by photosynthesis per square meter per day. And again, you can see lots of productivity on the west coast of the continents, yeah, and lots of productivity in the Arctic and the Antarctic. This is one reason why the whales go up to the Arctic to feed, and then they come down to Hawaii to breed. The phytoplankton, the krill feed on the phytoplankton, and then the whales feed on the krill. Okay, so this is where all the bioproductivity is. And again, the center of the oceans here is a desert. Now I want to show one more figure from De Maison and Moore, and this is going to be a striking difference you see. Now we're going to show you the organic matter that gets to the bottom and is preserved in the sediments. It doesn't look just like this, okay? It looks different. We'll have a look, and then we'll show you now. This is the weight percent TOC in the underlying sediments. Yes, there's a lot of that organic matter gets to the underlying sediments on the west coast of continents, right? We have these tremendous algal blooms, and then the algae die, and they start sinking through the water column. There's associated biological oxygen demand. The oxygen content drops down to 0 in the sediments, at least in the anoxic zone here, are anoxic, where you get preservation of the organic matter on the shelf. However, if you look at the Arctic and especially down here in the Antarctic, practically none of that organic matter, that high productivity, gets to the bottom and is preserved. There's essentially no leakage of organic carbon into the sediments here. Why is that? Well, the waters here are cool, they're cold, they're saturated with oxygen, 8 to 16 milligrams per liter, yeah, and there's a lot of oxygen in that water column, and there's not enough biological oxygen demand to use up all of the oxygen. So the bottom sediments are oxic. Okay, we're going to talk about 4 depositional anoxic settings. Yeah, and when you select your source rock, you want to see if they fit into one of these settings. Anoxic open ocean, it's pretty rare today, but it's characterized by global warming, usually a marine transgression, and it's widespread, the distribution of the anoxic sediments. They're widespread with usually little variation in the organophases. We have anoxic layers with upwelling, like I just showed you, offshore Namibia and Peru and California, low latitude shelves, the west side of continents where you have that Coriolis force. And these are typically narrow trends associated with phosphorites. When those organisms die, you know, they phosphorus is a limiting element that they require. They need phosphorus to survive, and when they die, that ends up in the sediments. And a lot of diatoms, at least since the Cretaceous, the diatoms have been a major contributor to sediments, especially in the Miocene. Anoxic sill basins. Yeah, these are temperate to warm, rainy, cratonic, or shelfal pockets. Commonly, they're richest in terms of the organic matter at the bottom, the center of the basin. And then we have anoxic lakes, equable, warm, rainy, usually equatorial. Early rifts is a typical setting, continuous within each lake system. Okay, so here's some modern and ancient examples of those settings. Anoxic open ocean, pretty rare today. You can find up in the northern part of the Indian Ocean, the bottom sediments are anoxic. And actually in the Northwest Pacific, it goes anoxic occasionally, mainly because of runoff from fertilizer from farming, but also there are other factors involved here. And you get these big fish kill-offs, you know, fish die-offs, okay? And the ancient example, of course, this is a huge source rock, the Cretaceous in the Atlantic. Okay? Ancient example. That was a early rifting of the Atlantic, and you had basically an anoxic ocean. Anoxic layers with upwelling. Well, present day, we gave plenty of examples of that. In the ancient environment, we're talking about the Miocene Monterey as one example. Yeah. And the Diacuran is another one in eastern Russia. The Permian Phosphoria Formation is another great example from central North America. Anoxic sill basins. Yeah, today we've got some of these like the Black Sea. I showed you some nice photographs of that. Baltic, Lake Maracaibo, Saanich Inlet. In the past, we had huge anoxic sill basins. There's a sill at one sort of the periphery of the basin that opens to the ocean. The Upper Jurassic in West Siberia, the Taurasian in the Paris Basin, not as great a class as this, but it certainly was anoxic. The Albion Maori Formation in central North America. And anoxic lakes, present day, you know, these are, the Tanganyika and Kivu are sort of equatorial lakes. They're characterized by thermoclines where that bottom water is isolated. In the Dead Sea, it's a halocline. Ancient examples, there's some great source rocks in the Cretaceous, the Songliao Basin, and also the Eocene and the Paleocene in China, and the Eocene Green River in central North America. So looking at the world's effective source rocks, I went to a paper by Klema and Gregory Omashek from 1991, and I just assigned I used their assignment of the oils to the source rocks by age, and I plotted it up as a pie diagram. The interesting thing here is that, you know, the world's source rocks are really biased in their distribution with respect to time. You don't see any significant Precambrian source rocks on here, do you? Well, Precambrian is a long time ago. And Precambrian oil, although we have billion-barrel fields in places like eastern Siberia, it's tough to preserve those accumulations worldwide over that long a time period. Here's some Cambrian-Ordovician source rocks here, okay? But the younger source rocks like the Aptian Turonian in the Cretaceous, 29%, that's a big chunk. The Upper Jurassic, those anoxic sill basins, like in West Siberia. There's also Upper Jurassic source rock in Middle East, the Hanifa Hadria in Saudi Arabia. And then, of course, we do have some older source rocks that represent large numbers. The Devonian, the Tornasian, and the Silurian in both South America and in North Africa. The Tanazuft, for example, in North Africa is a major source rock. But again, those are pretty old source rocks. The younger source rocks, we've got the oligomycene. Yeah, mycene's pretty young. They're pretty pronounced here. But this is a very biased look at the time the distribution of petroleum source rocks through time. So here's a source rock sample. There's some oils that might be generated from such a source rock. I want to spend some time in this early lecture to introduce the composition of petroleum. We can split petroleum using different chromatography techniques. Here we can take a crude oil, or we can grind up a rock, and we can extract the rock with a solvent, and then we can treat that using a short column chromatography. It's a good idea to de-asphaltene the oil first, because the heavy ends will plug the column. This really heavy material can plug chromatographic columns, and then we can split this into fractions using high performance liquid chromatography. So we can separate a saturated aromatic cut and a porphyrin or polar cut. And then we can split those with more with HPLC into saturated hydrocarbons. I'll describe those in a minute, and aromatic hydrocarbons. So a typical crude oil really has 4 major fractions. They're the asphaltenes, the heavy sort of ends. Those really haven't been studied that much in geochemistry because they're very complex. Okay. We then also have what we call NSO or polar compounds. NSO, nitrogen, sulfur, oxygen compounds, the polar compounds. Many of the porphyrins are there in the NSO fraction. And yeah, those have been studied a bit, but really a lot of the focus in geochemistry has been on these components, the saturated and the aromatic hydrocarbons. So asphaltenes, polars, saturated and aromatic hydrocarbons. Okay, and we separate those using high performance liquid chromatography, and there are lots of publications describing how that's done. Some academic labs use glass columns, alumina columns to do the separation, which is much more tedious and time intensive, but the automated way of doing it is with HPLC. Now, in terms of composition of petroleum, earlier I defined those 4 asphaltenes are not going to be on this figure from Bernard Tissot and Dietrich Welta, but I'm going to include the saturates, which are the alkanes and the cycloalkanes, and the aromatics and the NSOs. Now they've lumped those at this corner of the ternary diagram, and they have the alkanes here and the cycloalkanes there. So let's talk about some alkanes, just as an introduction here. This is the simplest alkane. It's methane. Methane, the carbon in methane is when it's the carbon is in an excited state, has 4 electrons that want to pair with electrons from other elements. So you want to form a covalent bond here that shares 2 electrons. And so that's a valence of 4. And so you have a tetrahedron here with methane, with a methane structure. And that's one way of showing it. CH4 is another, right? Now for normal alkanes, the next alkane in the normal alkane series is ethane. And you can see here C2H6. And again, the valence of 4 is satisfied in each case. Now here is another compound in the same homologous, we call it homologous series. It's a homolog, so it has an additional methylene group over and over again. This is a homolog in the series of N alkanes, and this one has 16 carbons in a row, and I'm not showing all the hydrogens, 3 here, 2 there, 2 there, etc. So this would be called hexadecane, N normal hexadecane. Now there are other alkanes, there are isoalkanes. You can have a methyl group here, that would be an isoalkane. You could have a methyl group here, that would be an anti-isoalkane. And then you have other branched alkanes. This is a compound, you see it's got 16 carbons in a row here, but it's got a methyl group every 4th carbon. So this would be called 2, 6, 10, 14 tetramethyl hexadecane. And sort of the common name for that compound is phytane. Phytane comes from the side chain in chlorophyll. Remember, chlorophyll is that photosynthetic pigment, and that's going to be found in virtually all sediments where you have chlorophyll being deposited with the organisms that are deposited there on those sediments and preserved. So that's phytane. That's a C20. Another series, it's a isoprenoid series, normal isoprenoid series, homologous series. Okay, let's look now at the aromatics. These are some aromatics. The simplest one would be benzene, and in benzene, there are only 6 hydrogens. You got 6 carbons and 6 hydrogens. So there are double bonds in here, like this. I'm showing you the double bonds. They move around. Those electrons are shared. So this is one way of showing shared electrons. So this is C6H6, right? And this is benzene. This is toluene with a methyl group, and then there are the xylenes, which have multiple methyl groups. So those are aromatics, and they're going to come out in the aromatic fraction. Here's an aromatic compound right here. This one is pretty interesting compound. This compound right here is let's see, that's benzopyrene or benzo-A-pyrene. That's a polycyclic, a polyaromatic compound. Very interesting compound. You find it in cigarette smoke. By the way, if you smoke, take some chemistry and quit. This compound right here is carcinogenic. If you go back to the Cretaceous Tertiary boundary, you can find a big spike right at the boundary in the ash layer, right at the boundary at Gubbio and other places, Gubbio in Italy and other places. And that's a big spike in this benzopyrene content. So that could mean one of two things. Either the dinosaurs were smoking a lot of cigarettes, or when that bolide impacted at Chicxulub in Yucatan, it generated a crater 200 kilometers or more across. It basically resulted in forest fires that burned more than 70% of the forests on the planet. The way this is a bad guy is that it's flat. If you look at it from the side, it's virtually flat. And in your DNA, adenosine, guanine, cytosine, thymine, right? Thymidine, this will fit right between those base pairs. So it can change the alphabet in your DNA. And 99.9% of the time, that's going to kill the cell, but every once in a while, that results in cancer. Not a good thing. Anyway, here's another compound over here. This will be classified as a polar compound. It's in the NSO, or the polar fraction. We call them NSO compounds. It's a little bit of a loose definition. This one you can see just has nitrogen. It's a tetrapyrrole. It's got 4 nitrogens in it and some oxygen. So it's got that one, but it doesn't have sulfur. Oh, it also has vanadium in there. So this is a vanadyl group in here. This is a porphyrin, and the precursor for this is chlorophyll, just like it is for the phytane. The phytane comes as a side chain that comes off of here as a phytal side chain, and that breaks off, and then this is left. There's usually a magnesium, well, there's a magnesium in here in chlorophyll, and that during diagenesis is replaced by a vanadyl group, or sometimes a nickel group, sometimes chromium or some other metal. Usually it's vanadyl or nickel. And so this is called a DPEP porphyrin, a deoxyphylloerythroetioporphyrin. R here can be all kinds of different functional groups. So this is a complex collection of compounds, actually. And it's difficult to study because there's so many of them and they're difficult to separate. But that is an NSO compound. Okay, let's look down here in this corner now and look at some of the examples we have of naphthenic compounds. Naphthenic means, they're going to be cycles. So here's a cyclohexane, C6H12. So it's got twice as many hydrogens as benzene, and it's a sort of a three-dimensional structure, a puckered ring. And that is, you know, there are a lot of different compounds related to that have functional groups, you know, methyl groups and ethyl groups around the side here. Here's a higher molecular weight cycloalkane. You can see it's a tetracyclic triterpene. It is a biomarker. And this one is, it's got a hydrogen at the 24 position. So it's got 27 carbon atoms in it. This would be cholestane. And cholestane, guess where it comes from? It comes from, it's a biomarker that comes from eukaryotes, from cholesterol. Cholesterol is a really vital cell membrane component. It helps to stiffen or rigidify the cell membrane and protect the cell from attack by other organisms. So this is highly resistant to biodegradation. And after the cell dies, the cholesterol becomes cholestane, the double bond is lost, the OH group is lost, and it becomes cholestane. In the average person, say 150-pound person, they're about 300 milligrams of cholesterol. But of course, if you eat too much cholesterol, too much fatty food, it will plaque out on your arteries and you can get high blood pressure and maybe even a heart attack because of that. So we manufacture cholesterol, we actually require it. Too much is not good for people. This is the last compound I want to give you an example of. This is a little ball here. You can see it's an unusual structure, isn't it? It's a little ball. It's got 10 carbon atoms in it. And you can see this is going to be thermally really very stable, because to break this apart, you're going to have to break at least 2 carbon-carbon bonds, and sometimes maybe even 3. Okay, this is a diamondoid. It's a diamondoid. It's in the class of compounds called diamondoids. And this particular compound is called adamantane. I used to work for a Mobil oil company, and we had a well in the Gulf of Mexico that tapped a reservoir that had smackover gas in it. It was very high mature gas. We thought it was pure methane. And when they opened the valve, they had a big ceremony to open the valve to the well. The gas started to come up the pipe and into the production lines. And then suddenly the gas flow stopped. And so they shut the well in, and they opened the pipe up, and they found the pipe was filled with this, for hundreds of meters, it was filled with this white powder, which was basically pure adamantane. So this was present along with pure methane, just pure methane, highly mature, along with adamantane. And as this gas with the adamantane came up through the pipe, the pressure and temperature went down, and this sublimated directly out of the gas phase as a solid. So there's just some examples of petroleum composition. We will talk a little bit more about petroleum later, but only in the context of what would be useful for you as a basin modeler. If you go into the field as a basin modeler or as a geologist, you probably want to take some sample containers with you, just in case you run across a sample that you want to collect from your study area. So, you know, glass bottles are great for oil samples. You want to have a Teflon liner here, because most caps, you know, the liner is a waxy material that is oil-based. And you don't want to fill this too much higher than the lip or the neck of the bottle here. And it's good to have a metal container, with a metal cap that fits snugly around the glass. Crude oils are commonly, they're emulsions. They have a lot of water in them, and it's good to store in glass, but if you stored it directly in the metal container, it would eat its way through the metal after a while. Glass, of course, breaks is easy to break, so it's good to have that metal container, snug fitting metal container along as well. Cloth bags are fine for collecting field samples of rocks. You want to evaluate a rock as a potential source rock. Again, don't mark the rock with a felt pen. That's organic. It's going to interfere with your chromatography, your geochemistry. Mark the bag. And also cores. These are great for samples. In most cases, it used to be that drillers could be fired for drilling and coring in source rock, That's changed, of course, with unconventionals. We want to know more and more about source rocks now. But a lot of rocks, especially those that have a reservoir interval in them, yeah, you might want to sample that to extract some oil if it's an oil-bearing reservoir. But many reservoir cores are preserved in wax. So they want to preserve the reservoir characteristics. And what wax is oil. So you don't want to deal with that. So avoid that. When you collect cores, and of course avoid core that's been marked with felt tip pens. Drill cuttings, well, that's sort of a last resort. Sidewall cores are sort of intermediate here. Drill cuttings, yes, many times they're fine, but you have to watch out with drill cuttings because, of course, they're easily contaminated with particulate additives, drilling additives. Many times you have little pieces of wood in there, or cigarette butts, or other particulate organic matter like lubber beads or plastic lubricant. The killer, though, the real killer, you can pick those out with a binocular scope. But the real difficulty here is when drill cuttings are contaminated with oil-based mud. And that, of course, makes geochemistry very difficult. It's best to get drill cuttings that have been drilled using water-based mud. Gas pressure vessels are fine. Teflon net for oil slicks. Sample handling is very important, but probably the most important item on this slide is this verbiage right down here at the bottom. I've seen more cases than I want to remember, really, where people have analyzed samples. They see some very interesting results and they say, gee, I'd like to look in more detail at that sample. Where's it from? And there's no recorded latitude or longitude, or the sample, the well name is not recorded, right? So every time you collect a sample or you have someone collect a sample for you, those people have to make very copious notes on the well name, the number, the operator, the latitude and longitude. If it's in North America, the API number, the depth, formation, what kind of sample it is. Is it a drill stem test, an RF, a repeat formation test? Is it a seep or a rock extract? All of that kind of information needs to be recorded to be useful. Now, as a basin modeler, you may not always have a geochemist handy to ask questions as you build your model. It's good to have geochemical input at various points in the preparation of your model, but geochemists may not always be there, so it's good to have these sorts of reference books available. These are just a few of what are out there, and the wonderful thing about these is they condense a lot of publications, thousands and thousands of individual published papers into one readily used volume. This volume, for example, the Biomarker Guide, has a nice glossary, and it's got a detailed index, so you can look up any parameter or any characteristic you want, not just biomarkers, but other things too, like rock eval and vitronite reflectance, and find out all the key references. So have these sorts of things available, not all of them, of course, but our BPSM PhD candidates at Stanford we have our own library with most of these types of books available for their use when preparing models. Okay, so some conclusions. We have 3 domains of life: the archaebacteria, the eubacteria, and the eukaryotes. These all contribute to sediments. The biomarkers that these contribute can help us to describe what the source rock was like, even if we don't have a sample of the rock. All we've got is the oil. Redox and sedimentation rate affect organic matter preservation. The relative rate of degradation of the total organic carbon can be addressed mathematically. We talked about Middelburg, and some of that is used, for example, in predictions of Methanogenisis. We will talk about that later. Oxic deposition favors bioturbated organic lean sediments. So you're looking for mud rocks, generally fine-grained rocks that were deposited under anoxic conditions. Some rocks that were deposited rapidly in deltaic settings can compensate for oxidation of the organic matter by getting that organic matter out of the oxic zone rapidly. But typical deltaic sediments usually don't exceed more than about 4 to even 5% TOC. Again, the Akbara formation in Niger Delta is no more than 4 or 5% TOC, dominantly terrigenous organic matter, of course. Anoxic settings, we've got 4 different anoxic settings. They favor laminated organic rich source rocks. So look for the lamination and see if you can fit your source rock candidates into one of these categories here. And remember that source rocks are unevenly distributed in space and time. You may have more than one source rock in your basin. Also, don't ignore that possibility. That's a key thing to keep in mind. And again, in the spirit of trying to keep this these lectures short and sweet and concentrated, I've really concentrated the references here. These are just some references you may want to have a look at. This paper, for example, by Hanneke Verwijs, is on prediction of microbial methane in the offshore of the southern North Sea, the southern part of the North Sea Delta. And this is basically a Petromod and biogenics modeling. It's a 3D model that was used to feed into 1D into biogenics to predict microbial or biogenic gas generation. Okay, some additional references. Well, you're a basin modeler, so you know about these references, but just in case, I'm listing them here. And of course, this book by Thomas Hanschel and Armin Kaurauf is a classic and is a very useful reference. Even for experienced basin modelers, it's a good thing to go back and look at the details of what's involved in some of these modeling algorithms that are used. I also have some CDs and some papers out on this topic. Now, on another topic, I just want to make a little suggestion here at the end. We're all either directly or indirectly involved in the petroleum industry. This is the petroleum age. If you look at terms of geologic time, the petroleum age is not going to be even a little blip. It's a very short-term thing, lasting no more than a few hundred years, right? Petroleum is a finite resource, like so many other things. It's going to be around for at least a couple hundred years, that's for sure. There's going to be a need for good petroleum geochemists, good petroleum basin petroleum system modelers, but we're going to have to make a switch, gradual switch. It's going to take time and it's going to take a lot of planning. Present day, about 85% of our global energy demand is satisfied by fossil fuels. That's not going to change very much in the near future. It's going to be many decades before we get other alternatives that can start to make a dent in the huge amount of energy that we're using on a daily basis. That's fossil fuel. But this is a very good discussion of the switch that's going to have to occur. It'll take you about 1 hour and 40 minutes to go through it. This was provided by the Bureau of Economic Geology at the University of Texas at Austin. Scott Tinker was directly involved in making this, and it's very entertaining and I think very informative. So as a geologist, as a basin modeler, as a geochemist, I think it would be beneficial for everybody to have at least had a look at this. All right, well, I've said pretty much what I want to say about the origin and preservation of organic matter. Now we're going to get into vitrinite reflectance. And this next lecture is not sort of your run-of-the-mill talk about vitrinite reflectance. I'm going to pick out and discuss some of the most recent developments in vitrinite reflectance. I'm going to talk about some of the limitations and the pitfalls in interpreting reflectance. It's unfortunate that a lot of this information is sort of lost in translation between the geochemist and the basin modeler. And I want to make sure that basin modelers are aware of all the new developments here, developments in the reflectance measurements itself, developments in the kinetics of calculating reflectance, and so forth. This will be discussed also in the kinetics section a little bit. But the next one will be on vitrinite reflectance, and I look forward to having you attend that and listen to that. Now, at the end of each lecture, I'm going to add a little homework assignment. This is a little exercise you might want to think about, and we will discuss it in the next lecture. This is sort of a composite depth versus vitrinite reflectance plot. And I'm basically going to ask you to think about what is happening in each of these intervals here, for example. What's happening right here between D and E? Yeah, and how does that affect your model, your basin model? What's the thermal maturity in this zone right here? Is it this or is it over here? Yeah, what's going on up here? What's going on at this dog leg right here? Is that because of a change in the geothermal gradient, or is there something else that's causing that? So this is a little homework. Think about that. Well, at the very end here, I've got you know, a little information about myself. I think most of you probably know who I am. I've got many years of experience in organic geochemistry, petroleum geochemistry, and in basin modeling. And I'm just one of many now co-principals in our BPSM program at Stanford. We have a tremendous breadth of knowledge among those co-principals, and our students are benefiting from that. And so I'm hopeful that my co-principals will also present similar lectures to the ones that I will present here, these 6 lectures that I'm going to present here. And I know that those will be of value to you as well. So I'm looking forward to seeing you at the next lecture. That's lecture number 2. Thanks for attending.